Today, we will be using magnetic nucleotides to translate a portion of a gene using the components included in Carolina Biological's modeling DNA to protein kit. We will need an mRNA sequence made of magnetic nucleotides, a ribosome transparency, a pool of amino acid beads, a section of pipe cleaner, tRNA magnets, and a magnetic whiteboard. Using magnetic nucleotides, we have modeled a transcript of messenger RNA, or mRNA, from the instructions found within a sequence of DNA nucleotides. During the process of protein synthesis, or translation, the genetic code found within a sequence of mRNA nucleotides is translated into a sequence of amino acids called a protein. This magnetic nucleotide series represents a sequence of codons for a region of a gene. Codons are groups of 3 mRNA bases. Each codon signifies a specific amino acid during protein synthesis. The first step of translating a mRNA sequence into a protein is initiation. Follow these steps to model the process of initiation. The codons do not start at a random site. The first mRNA codon is always AUG. Locate the AUG codon and then place the P site of the ribosome transparency over this start codon. The order of the nucleotides in mRNA determines the specific order of amino acids incorporated into the growing protein chain. Use the mRNA codons for amino acids chart to determine the amino acid that matches the AUG codon. Note, the base pair combinations for each of the 20 amino acids are followed by the name of the amino acid and, in parentheses, its 3-letter and 1-letter abbreviations. Using RNA base pairing rules, A pairs with U and C pairs with G, locate the tRNA magnet containing the complementary RNA sequence to the AUG start codon. During protein synthesis, tRNA molecules carry specific amino acids from the cytoplasm to their matching mRNA codon. These amino acids will then be transferred to the growing protein chain, represented by a colored pipe cleaner, as it elongates. Your instructor has designated an area to serve as the cytoplasmic reservoir of unbound amino acids. Carry one pipe cleaner and the tRNA magnet corresponding to the AUG start codon to this amino acid reservoir. The purpose of the transfer RNA, tRNA, is to move the correct amino acid to the growing protein chain. Use the codon chart to locate the bead inscribed with the 1-letter abbreviation of the amino acid that corresponds to the AUG start codon, and then thread the amino acid onto the pipe cleaner to simulate the placement of the first amino acid in the growing protein chain. Place this tRNA amino acid complex on the P site of the large ribosomal subunit. It may help to bend the bottom 4 centimeters of the pipe cleaner to form an L to achieve a better fit when using the model. The mRNA codon located at the current A site, attachment site of the ribosome, specifies the next amino acid to be added to the growing chain. Locate the tRNA magnet that corresponds to the mRNA codon at the current A site of the ribosome, and then carry this tRNA to the amino acid reservoir. Remember, the mRNA codon occupying the A site of the ribosome, not the tRNA anticodon, specifies the correct amino acid. Using the codon chart for this example, the mRNA codon, GUG, codes for the amino acid valine, which is abbreviated as V. Retrieve the amino acid and then place the appropriate tRNA magnet and its corresponding amino acid on the current A site of the ribosome. Move the pipe cleaner representing the growing amino acid chain from the P site to the A site. Transfer the amino acid corresponding to the tRNA found at the A site to the growing amino acid chain to simulate a peptide bond, linking the amino acids together. Hold the tRNA magnets in place and then advance the ribosome transparency a distance of 1 codon to the right direction on the magnetic mRNA sequence. The current amino acid chain should reside on the tRNA found in the P site. The transfer RNA that carried the first amino acid in the sequence, methionine, or M, is now released from the E site and may be reused. Simulate this event by removing the magnetic tRNA from the E site. The next tRNA may now move into the now vacant A site position, carrying with it the appropriate amino acid that corresponds with the A site codon. Repeat the process of protein elongation using the appropriate tRNA magnets and amino acid beads. Important: remember to advance the ribosome transparency a distance of 1 codon after transferring each amino acid to the growing protein chain. Elongation of the protein chain is terminated when a stop codon moves into the A site of the ribosome complex. A stop codon does have a corresponding tRNA. It does not specify an amino acid. A protein called a release factor now binds to the stop codon located at the A site. To simulate this event, place the release factor magnet over the A site. When the release factor occupies the A site, the completed protein, aka the amino acid chain, may be released. Separate the newly translated portion of the protein chain represented by the pipe cleaner containing a sequence of amino acid beads from the tRNA located at the P site and remove the ribosome transparency from the mRNA strand.